Welcome back to the deep dive. If you've ever had to grapple with complex cancer biology, you know that pancreatic ductal adenocarcinoma, PDAC, is, well, it's the definition of a stubborn adversary.
Absolutely. It has that devastatingly low 5-year survival rate, doesn't it? Hovering around just 12%.
Exactly. And when it spreads, when it metastasizes, we often just assume the new tumor is basically a copy of the first one, maybe with a few extra glitches thrown in.
Right, the standard thinking. But the sources we're diving into today, this really is an unprecedented biological atlas. They prove that assumption is, well, fundamentally flawed.
Flawed how?
That's the key takeaway right there. For decades, research focused heavily on genomics, the cancer cell's sort of fixed DNA blueprint.
The mutations, the basic code.
Exactly. But we've come to realize that metastatic cells don't just carry that blueprint. They seem to carry a kind of survival manual that changes based on where they land. And this new study gives us the first really comprehensive look at transcriptomics. That's the active gene expression. You could think of it as the cell's current job description, and crucially, it looks at it in a spatial context.
Okay, so our mission today is to understand how this spatial map changes everything we thought we knew about PDS metastasis.
Yeah.
They use something called spatially resolved transcriptomics, SRT.
Yep, as of
On 55 samples. These were primary tumors and metastases collected from 13 patients. And the collection method itself sounds important, this rapid autopsy approach. Why was that so groundbreaking?
Oh, hugely groundbreaking, because usually metastatic tissue is sampled piecemeal, bit by bit, often after it's been sitting in cold storage for ages, which can degrade the RNA you need for transcriptomics.
Right.
These researchers used warm, rapid autopsy samples that allowed them to get high-quality matched primary tumors and critically synchronous metastases from multiple different organs all at once: the liver, the lung, the peritoneum.
So like a snapshot in time across the whole disease landscape.
Exactly, a synchronized snapshot of the disease's evolution across the body, right at its most aggressive stage. And what jumped out immediately? The most dramatic differences in how the cancer cells were behaving, their transcriptomics, were found right between the liver metastases, limb, and the lung metastases, limb.
Okay, liver versus lung. Let's dive into that. So starting with those functional changes, the job description, as you put it. If a cancer cell starts in the pancreas, then travels and sets up shop in the liver, how does its gene expression profile actually change?
Well, first off, tumors from the same patients still shared core similarities. That makes sense, right? Confirms a common origin.
Okay, so they're related.
Definitely related. But once you look past that baseline similarity, the local organ environment, the niche, exerts this powerful selective pressure. The cancer cells develop really quite substantial transcriptomic differences based on where they metastasize.
And you mentioned that striking contrast between liver and lung, especially in patients who hadn't had treatment yet.
Yes. In those treatment-naive patients, the liver metastases, the limb, were heavily focused on, let's say, structural adaptation. They showed increased activity in pathways linked to the extracellular matrix, the ECM.
The ECM, that's like the scaffolding between cells.
Exactly, the physical scaffolding and glue holding tissue together. So the liver tumors were basically busy building this dense, rigid architectural network around themselves. Very structural.
Okay, and the lung.
The lung metastases were quite different. They seemed more focused on engagement, interaction. They were enriched for immune-related signatures. Things like T helper cell differentiation were more active. It suggests the lung site was inherently more interactive with the local immune system from the get-go.
Interesting. That sort of makes anatomical sense, maybe. But the treatment data, that's where you said it gets really surprising. What happens when you give systemic chemotherapy? Do those strategies just get shut down?
This is where it gets really interesting, because the pattern didn't just fade, it shockingly reversed.
Reversed? How so?
In patients who had been treated, those ECM-related signatures, that dense scaffolding activity, were now enriched in the lung metastases.
So the lung tumors started building the fortress after treatment.
Right. And meanwhile, the immune-associated features, things like granulocyte activation, they became more prominent in the liver metastases post-treatment.
Wow. A complete flip. That suggests treatment isn't just killing cells, it's acting like a selective pressure cooker, isn't it?
Precisely.
It's forcing the surviving cancer cells in the liver and the lung to switch their survival tactics entirely. It's like the therapy unintentionally trained the cancer to learn two completely different resistance tricks, depending on its location.
That really highlights the challenge of systemic therapy, doesn't it? The effective treatment is inherently niche-specific. You can't assume it works the same way everywhere.
Makes sense.
And the study also noticed something else interesting. The primary tumor often seemed transcriptionally closer to peritoneal metastases, those in the lining of the abdomen, than to the liver or lung tumors.
What does that suggest?
It suggests that colonizing the liver and the lung might require a more distinct, maybe a more specialized evolutionary step compared to spreading within the abdominal cavity. They're just tougher neighborhoods to crack, perhaps.
So location changes the job description, and treatment forces a complete rewrite depending on that location.
Yep.
But what about the underlying genetics? If the function changes so dramatically, how does the cancer's DNA, its family history, keep up? Let's talk clonal evolution. How do they map the genetic changes?
Right, so they moved from the transcriptomics to the underlying structure, the genomics. They used a specific algorithm, it's called spatial infer CNV.
Spatial infer CNV.
To look at copy number variations, or CNVs. These are those large-scale gains or losses of chunks of chromosomes. They're hallmarks of cancer's genetic instability. By mapping these CNVs spatially, they could basically build a phylogenetic tree, like an ancestry map for each patient's cancer, showing how different clones evolved and spread.
Okay, so they built family trees for the cancer cells in each patient. What did those trees reveal? Was there a single predictable route for how PDA spreads?
Predictable. No, the central finding was, well, chaos is maybe too strong a word, but extreme diversity. There was no single dominant pattern of dissemination across all the patients.
So every patient's cancer spread differently.
Pretty much. It reinforces that PDA's evolution is highly patient-specific, highly diverse. They did manage to categorize this diversity into several patterns, but maybe two stand out as particularly counterintuitive, highlighting just how adaptable this cancer is.
Okay, give us the highlights. What were those surprising patterns?
Okay, one was what they called parallel evolution or expansion. Picture this. You have a clone, a specific genetic subtype that's only a tiny minor population in the original primary tumor, almost insignificant there.
Right.
But then once cells from that clone reach the liver, suddenly, bam, it becomes the absolute dominant clone there. It completely takes over the liver metastasis.
Whoa. So a background player in the primary tumor becomes the star in the liver.
Exactly. It suggests that the specific CNV profile carried by that initially minor clone, maybe it had gained a specific gene or lost one, gave it the perfect selective advantage it needed to survive and absolutely thrive only in the liver's unique environment. It wasn't needed in the pancreas, but it was the key to the liver.
So the primary tumor is like a crowded bus with lots of slightly different passengers, and the liver is a specific job site looking for one very particular genetically unique construction worker from that bus. Fascinating. Okay, what was the second really surprising pattern?
That would be what they termed divergent patterns within synchronous metastases. Here, they looked at patients who had two different metastatic lesions, two tumors, in the exact same organ at the same time, say two separate spots in the liver.
Okay, neighbors in the same organ.
Right. And even these two neighboring lesions could have dramatically different clonal compositions. They might have evolved differently from the primary tumor and differently from each other.
Even within the same liver.
Even within the same liver. It just shows the sheer power of heterogeneity, the diversity, happening even at a micro-regional level within the same host organ. The local pressures can be that specific.
Incredible. So the genetic roadmap, the clonal evolution is clearly important, and they did validate some of this, right? Like showing that successful metastatic clones often had known advantages, like higher amplification of the MYC oncogene.
Yes, they did. They used Phi-SH fluorescence in situ hybridization for MYC on chromosome 8. And indeed, they confirmed that in some patients, the clones that became dominant in the liver or lung metastases had higher levels of MYC amplification compared to the primary tumor clone. That fits the idea of selection for advantageous mutations.
Okay, so genetics do matter for providing the raw material for selection. But if the genetics, the clonal evolution, is so variable and almost chaotic between patients, and even within the same organ, how can the function, the job description we talked about earlier, be so relatively uniform? You know, the liver tending towards ECM production, the lung towards immune interaction, especially before treatment. That seems like a contradiction.
You've hit on it. That question leads us directly to what I think is the study's real aha moment. It's this concept of cancer cell lineage states.
Lineage states, what are those?
Think of them as cellular identities or phenotypes. They defined three main ones based purely on gene expression profiles on the transcriptomics, regardless of the underlying genetic mutations or CNVs. There's the classical state, generally considered less aggressive. There's the basal-like state, which is known to be highly aggressive, fast-growing, invasive, kind of the pioneer identity, and then an intermediate state in between.
So classical basal-like intermediate, defined by how the cell's behaving, not its deep genetic code. How did these identities map onto the different metastatic locations?
This is where it gets really clear. There was a distinct, quite dramatic organ-specific lineage shift. The liver metastases and also the peritoneal metastases showed a really noticeable predominance of that highly aggressive basal-like state.
How predominant?
About 75% of the cancer cells in the liver samples showed a basal-like signature, and they had minimal classical state cells.
Okay, liver favors the aggressive basal-like identity. What about the lung?
The complete opposite, essentially. The basal-like state was substantially underrepresented in the lung metastases. The lung environment seems to favor colonization by cells adopting the less aggressive classical state.
They confirmed this somehow.
Yeah, they validated this using mouse models, KPCY mice, which spontaneously develop pancreatic cancer and metastases. They stained the tissues for specific markers. S100A2, which is high in basal-like cells, and GATA6, high in classical cells, and the pattern held up basal-like preferred the liver, classical the lung.
Okay, wait. I need to pause on the significance here because this sounds huge. We just talked about the chaotic clonal evolution, the different CNVs, the MYC amplifications, that's the genetics. Isn't cancer fundamentally defined by its mutations? Are you seriously telling me that the underlying DNA changes, the clonal ancestry, don't matter as much for the cell's behavior as the neighborhood it lives in?
That is precisely the groundbreaking conclusion they reached. The lineage diversity, whether a cell acted basal-like or classical, was largely independent of the cell-intrinsic clonal architecture.
Wow.
You could literally find two cancer cells sitting side by side in the tissue, having the exact same genetic CNV profile belonging to the exact same clone. Yet one exhibits the aggressive basal-like gene expression pattern, and its neighbor remains in the classical state.
So the local environment, the microenvironment, is dictating the cell's identity, its lineage state, its fate, almost overriding its fundamental genetic programming.
That's what the data strongly suggests. The microenvironment seems to be the dominant force shaping the functional state, the lineage identity of the metastatic cell once it arrives.
That completely reframes how we should think about metastasis. If the microenvironment is the ultimate driver of this aggressive basal-like state, what functional characteristics come along with that identity? What makes it so bad?
Well, when a cell flips into that basal-like identity marked by genes like S100A2 and KRT17, it strongly activates several really adverse functional programs. We're talking hypoxia response.
Thriving in low oxygen.
Right. Also, epithelial mesenchymal transition, or EMT.
Becoming more invasive, migratory.
Exactly. And MYC programs driving proliferation. These basal-like cells also tended to be most prevalent right in the tumor core, the center of the metastatic lesion.
Suggesting they're the specialized survivors, maybe, thriving in the harshest, most oxygen-deprived part of the tumor.
That's a very plausible interpretation. The core is a tough place to live, and the basal-like state seems well-equipped for it. Meanwhile, the intermediate spots often showed links to things like interferon responses and MHC class II programs suggesting more immune interaction there.
Okay, so if the microenvironment is driving this aggressive basal-like identity, we absolutely have to look closely at the neighbors, the non-cancer cells in that environment. They use some high-resolution mapping techniques, right? Like iStar.
Yeah, iStar, which gives you super-resolution gene expression mapping. It lets you see not just what genes are on, but precisely where they are on and which cells are nearby. This allowed them to map out the tumor microenvironment, the TME, in incredible detail.
So which non-cancer cells were these aggressive, basal-like pioneers collaborating with most closely? Who are the main accomplices?
The data pointed overwhelmingly to one specific cell type, the myofibroblastic cancer-associated fibroblasts, or myCAFs.
myCAFs. Okay, what are they normally?
Think of them as activated fibroblasts, kind of like specialized scar tissue construction workers. They produce a lot of extracellular matrix. And these myCAFs showed this incredibly tight spatial co-localization almost exclusively with the aggressive basal-like lineage state cancer cells.
They were hanging out together. Where exactly were these myCAFs stationed and what were they building there?
They were definitely enriched throughout the tumor bed where the basal-like cells were, but their really crucial role seemed to be in the juxtalesional regions.
Juxtalate.
Meaning the stroma, the connective tissue, immediately adjacent to the edge of the tumor cluster. And there, the myCAFs were often forming this dense, almost continuous band, literally encapsulating the cancer foci, like building a physical defensive wall around the basal-like tumor cells.
A wall built by these myCAF cells. What signals them to build it?
The study suggests this construction is likely orchestrated, at least in part, by the cancer cells themselves. Specifically, areas rich in basal-like cells and myCAFs also had high levels of TGF-B1 expression. TGF-beta1 is a well-known cytokine that promotes myCAF differentiation and, importantly, is also strongly immunosuppressive.
Okay, so the basal-like cells might be pumping out TGF-B1, which tells the myCAFs to build this dense, protective, immunosuppressive wall around them. How effective was this wall at keeping the immune system out? PDCAS isn't known for high immune infiltration anyway, right?
Right. It's generally considered an immunologically cold tumor. But even within that context, this myCAF barrier seemed highly effective at exclusion, particularly against one key immune cell type, plasma cells.
Plasma cells. Those are the antibody factories, right? Part of the humoral immune response.
Exactly. Critical for producing antibodies that could potentially target cancer cells. And these plasma cells were almost entirely excluded from the tumor bed itself. They were stuck outside that myCAF barrier in the juxtalesional zone.
Wow. So the wall specifically keeps the antibody producers out. Is there a known mechanism? How does the myCAF barrier actively lock the plasma cells out? What's the key in the lock?
This is the final, really compelling piece of the mechanism they uncovered. It seems to operate via the CXCL12-CXCR4 axis.
Okay, break that down. CXCL12 and CXCR4.
So the myCAFs, those wall-building cells right at the border, were found to be expressing high levels of a specific cytokine, a signaling molecule called CXCL12.
Okay, myCAFs make CXCL12.
Then, using even more advanced spatial profiling, a technique called COSMX, which looks at single cells in situ, they confirmed that the plasma cells, which were stuck just outside the barrier, predominantly expressed the receptor for CXCL12. That receptor is CXCR4.
So the myCAFs put out the CXCL12 signal, and the plasma cells have the CXCR4 receiver antenna.
Precisely. And when CXCL12 binds to CXCR4, it can act almost like a chemical anchor or a chemoattractant that holds the cells there. The fact that they saw CXCL12 expressing myCAFs directly co-localized with CXCR4 expressing plasma cells right at that barrier strongly suggests that this specific signaling axis is actively retaining the plasma cells in the stroma, physically preventing them from breaching the wall and entering the tumor core to do their job.
It's like a chemical fence powered by the myCAFs, specifically designed to keep the plasma cells out.
That's a great way to put it, a chemical fence reinforcing the physical myCAF barrier.
This really completes that organ contrast picture then, doesn't it? The low metastases, which favored the less aggressive classical lineage, didn't seem to build this intense myCAF CXCL12 barrier. They had more immune infiltration overall. T cells, B cells, plasma cells, other types of SAFs like APCFs involved in antigen presentation, and they had lower levels of that immunosuppressive TGF-B1. They were more visible. Whereas the liver metastases, favoring the aggressive basal-like lineage, they specialized in building this invisible fortress using the myCAFs, fueled by TGF-B1, and employing that specific CXCL12, CXCR4 lock and key system to achieve immune exclusion, especially of plasma cells.
That's exactly. The location dictates the lineage state. The lineage state determines the specific architecture of the tumor microenvironment, including collaborators like myCAFs. And that TME architecture, in turn, controls immune access and ultimately contributes to therapeutic recalcitrance. The whole chain reaction laid out spatially.
This has been a genuinely deep dive into the spatial complexity of PDHE metastasis. I mean, the main takeaway seems crystal clear. We absolutely cannot treat liver metastasis as functionally or structurally identical to lung metastasis anymore. The location, the address, profoundly shapes the cancer cell's identity and behavior, seemingly overriding even its core genetic instructions.
And the clinical relevance here is just massive, isn't it? If the most aggressive basal-like tumors, which are predominant in the liver, the most common site of PDA metastasis, if they rely so heavily on this physical myCAF barrier, and specifically on the CXCL12, CXCR4 axis for immune evasion, well, now we have precise, tangible targets.
Targets beyond just the cancer cell itself.
Exactly. We need to move beyond only targeting the cancer cell's DNA or growth pathways and start thinking about how to dismantle the physical and chemical barriers that the microenvironment creates around the cancer. And crucially, this CXCL12, CXCR4 axis can be pharmacologically targeted. There are drugs in development or use for other conditions that hit this pathway.
So this spatial map provides potential immediate therapeutic avenues.
It really does. It underscores the need for this nuanced spatial understanding to develop more personalized, effective interventions. Maybe we need liver-specific strategies targeting myCAFs or CXCL12, particularly for those basal-like tumors.
Which leaves us with a really provocative question for you, the listener, to think about. Since the evidence suggests the cancer cell's lineage state, its functional identity, is largely driven by the microenvironment, not just its genetics. And we now have a detailed map showing exactly how the microenvironments of the liver and the lung differ. One builds that protective basal-like fortress with myCAFs and CXCL12. The other maintains a more classical immune interactive state. How could we potentially exploit those very different environmental conditions? Could we design two entirely different organ-specific treatment strategies based not just on the cancer's mutations, but on the unique vulnerabilities created by its metastatic address? The future of oncology might really depend on knowing not just what the cancer is, but exactly where it is.